Hi, I am Dr. Sandeep Nagre. First of all, heartfelt congratulations to Kendric Cromer, along with the dedication of scientists, clinicians, and researchers, exemplifies the power of collaboration and innovation in transforming lives with the remarkable achievement in the field of gene therapy. Kendric Cromer, a brave 12-year-old from Washington, D.C., has made history as the first person outside of clinical trials to receive Lyfgenia, a groundbreaking one-time gene therapy for sickle cell disease. This milestone represents a profound leap in medical science and personalized care, offering hope for countless individuals living with this challenging condition. This achievement is not just a victory for Kendric, but for the entire global healthcare community, as we witness the potential of cutting-edge therapies in addressing genetic disorders. Let's talk today about this truly revolutionary topic, gene therapy, and its promise for patients with sickle cell disease. Sickle cell disease, or SCD, is a genetic condition that affects millions of people worldwide. It affects red blood cells. Instead of the normal round shape, the red blood cells become stiff, sticky, and shaped like sickles. This abnormality blocks blood flow, causing severe pain, organ damage, and even premature death. Despite the progress in managing symptoms, treatments remain limited, and most aim to relieve symptoms rather than address the root cause. But now, with advances in gene therapy, we're moving from managing symptoms to potentially curing the disease. Treatment, which is making significant strides, is Lyfgenia and Casgevy. Gene therapy involves altering a person's DNA to treat or potentially cure diseases. In the case of sickle cell disease, this therapy can target the gene responsible for producing the faulty hemoglobin that causes sickling by editing or replacing the defective gene. Gene therapy offers the possibility of normalizing red blood cell function and essentially curing the disease, using advanced techniques to introduce a functional copy of the gene that codes for normal hemoglobin. This enables patients' bodies to start producing healthy red blood cells again. Lyfgenia's gene therapy starts with collecting the patient's own stem cells. These cells are then modified in the lab to carry the correct version of the hemoglobin-producing gene. Once the cells have been edited, they are reintroduced into the patient's bloodstream. Over time, these corrected cells multiply, leading to the production of healthy red blood cells, reducing or even eliminating the complications associated with sickle cell disease. What makes Lyfgenia's approach unique is their use of advanced viral vectors, ensuring that the therapeutic genes are delivered efficiently and safely into the patient's cells. This innovation not only enhances the success rate, but also minimizes risks, making gene therapy a more viable option for a broader range of patients. So, how is it working in real life? Early clinical trials of Lyfgenia's gene therapy have shown incredible promise. Many patients who previously suffered from frequent pain crises and organ complications have reported dramatic improvements, some even becoming symptom-free. For patients with sickle cell disease, this kind of therapy could mean a life without daily medications, blood transfusions, or the constant fear of severe pain. This is particularly life-changing in regions where access to traditional therapies is limited. Next up is Casgevy, another cutting-edge approach, but this one focuses on gene editing rather than gene addition. Casgevy uses the CRISPR-Cas9 technology to edit the patient's DNA directly. Here's what makes Casgevy stand out. Instead of adding a new gene, this therapy corrects the patient's own DNA. The CRISPR-Cas9 system is programmed to target the BCL11A gene, which suppresses the production of fetal hemoglobin, a form of hemoglobin that doesn't cause cells to sickle. By editing this gene, Casgevy allows the body to produce fetal hemoglobin, which compensates for the defective hemoglobin S and helps prevent the sickling of red blood cells. The results so far have been incredibly exciting. Patients receiving Casgevy in clinical trials have shown significantly reduced symptoms and fewer complications. And like Lyfgenia, some patients are becoming less reliant on blood transfusions, and their quality of life has improved dramatically. What's especially promising about Casgevy is that it offers a more targeted, potentially one-time treatment, as opposed to the ongoing management that sickle cell patients have historically faced. Of course, there are still challenges to address. Gene therapies are complex and expensive, which limits access to patients who might benefit from it the most. However, companies are working to streamline production, reduce costs, and make these therapies more widely available. Additionally, ongoing research aims to make gene therapies safer and more effective, with a focus on refining the gene editing techniques to avoid unintended consequences. But what's undeniable is that gene therapy represents a monumental shift in how we think about treating genetic disorders like sickle cell disease. The future is bright, and it's thanks to innovations like Lyfgenia and Casgevy. The impact of gene therapy on sickle cell disease could be revolutionary, not just for those affected by the disease, but for the entire field of genetic medicine. Lyfgenia and Casgevy is leading the way, pushing the boundaries of what's possible and giving hope to patients who once faced a lifetime of suffering. Thank you for tuning in. If you enjoyed this episode or found it informative, be sure to subscribe, leave a comment, and share it with others who are passionate about the future of healthcare. Until next time, stay curious and stay hopeful.